Okay, hello Bio2. This is Mr. B and we're continuing on with Chapter 23. We just finished Section 2 about roots, so now we're going to get in Section 3 and that's going to deal with stems. So to start, we're going to look at what are the structure and function of stems. So starting with the function. They have three important functions. They produce leaves, branches, and flowers. They hold the leaves up to the sunlight, probably carry out photosynthesis, it's very important, and they transport substances between roots and leaves, and that would be of our vascular tissue, of our xylem, and our phloem. So there are three most important functions of a stem. Produce leaves, branches, and flowers, hold leaves up to the sunlight, and transport substances between roots and leaves. And some other things about their function, they make up an essential part of the water and mineral transport systems of the plant. I mentioned that already. The xylem and phloem form continuous tubes from the roots through the stems to the leaves. So they're just kind of starting in the roots, through the stems to the leaves. If they didn't have the stem in there, they'd have no way of getting from the roots to the rest of the body. And this allows the water and the nutrients to be carried throughout the plant. So that is the main functions of our stem there. Big one being that bridge between the roots and the leaves so we can get some growth going on in our plant. So we're going to look at the structure of a stem. Stems, to start, are surrounded by a layer of epidermal cells that have a thick cell walls and a waxy protective coating. So this is just to protect the stem because, again, it needs to hold up a lot of different things, or maybe not a lot of different things, but a lot of the same thing, a lot of leaves to help the plant reproduce and to photosynthesize. Sorry, not to reproduce, just to photosynthesize. So that's why it needs to be strong and have that thick cell wall and then a waxy protective coating. And we got a nice little image here. Leaves are attached to the stem. Its structure is called nodes. So over here on your right, you can see where the leaf comes into the brown part of the stem at the area called the node. And the region of the stem between the nodes are called internodes. So from one leaf right here to this other leaf right here, that whole area is called an internode because it is in between two nodes, so that is a pretty self-explanatory name there. And then small buds are found where leaves attach to nodes. So you can see, well, the one without the line, you can see it a little bit better how there's a little growth going on right there, and that is being known as the bud. So this is some of the structure of the stem. The buds, they contain undeveloped tissue that can produce new stems and leaves, and in larger plants, stems develop woody tissue that helps support leaves and flowers. So that would be like our trees and our shrubs and things like that. So that is some of the basics of the stem structure. What did we have? We had node, internode, bud, and that was it. And then just know that in larger plants, the stems develop a woody tissue that helps support leaves and flowers. So now we're going to take a more in-depth look at the differences between the stems and monocots and dicots. And we're going to look at how the arrangement of the tissues inside, and arrangement is misspelled here, I'll take care of that, arrangement of tissues in the stems differs among seed plants, and those two seed plants being monocots and dicots. So in monocots, vascular bundles are scattered throughout the stem, so our xylem and our phloem are scattered throughout the stem in monocots, and in dicots and most gymnosperms, vascular bundles are arranged in a ring-like pattern. So we'll see exactly what that looks like. But first we're going to look at the monocots. So they have a distinct epidermis, so that just means a distinct outer layer which encloses the vascular bundles, and each vascular bundle contains xylem and phloem tissue, which we all know that by now because we've been talking about plants for a while. We know xylem and phloem make up the vascular tissue. So here is a cross-section of a monocot stem. So we can see that the vascular bundles are scattered throughout the ground tissue. So each one of these little circles in here are a vascular cylinder, our xylem, and our phloem. Our vascular bundles, they're labeled here. And then we can see the epidermis is going to enclose each one of those vascular bundles. And then the ground tissue consists mainly of parenchyma cells. So we can see the rest of this filler area. Anything that's not a circle is going to be our ground tissue made of parenchyma cells. And that is it for the monocot cross-section. So now we're going to look at the cross-section of a dicot. And as we can see in the dicot stems, they have vascular bundles are arranged in a ring-like pattern. So these bigger globs out here with the purple and the blue, those are our vascular bundles. As you can see, there's none in the center. They're all in a ring-like pattern going around the outside. And then the parenchyma cells inside the vascular tissue, for some reason, are known as pith. That's just what they're called. So this filler material inside of here is known as the pith of the stem, and the vascular bundles go around the outside. And so that is it for this one. If you have any questions, let me know.